Hello, everyone. I'm Daria, and today I'm going to be talking to you about the visual system. So, starting off, we as humans can only see certain lights in certain wavelengths. Our wavelengths range from about 380 nanometers to about 750 nanometers. And this is what we call the visible light. This is what we can see, and these wavelengths correspond to specific colors, as you can see here. And this, out of all the wavelengths, we see a very small amount, but it's interesting to think about what our view of color is and the different wavelengths in comparison to dogs' views, which, as you can see, dogs can only see two colors really, blue and yellow, and cats are different as well. So it's kind of interesting to look at what we can see compared to other species as well. And it's really cool because we can see a lot of color compared to certain animals. So looking at the anatomy of the human eye, there's a lot to it, and we won't go super in depth into the whole eye, but here we can see like the muscles that are attached that allow us to move our eye to and the eyelid and the eyelashes, which are a great protective barrier to our eye. But we're going to focus on how light enters the eye and the main structures that are part of that system. So light enters through the eye and it goes through the cornea here, and it is focused by the lens. And the lens then inverts the visual field onto the retina. So kind of goes back and lands on the back of the eye. This kind of like red area is the retina, which is a very important structure and is where our photoreceptors are. And the retina here then leads to the optic nerve. So the neurons kind of converge together and lead to the optic nerve. And the optic nerve then kind of goes back to our brain and is able to be processed there to go on to have different functions. Um, so looking at the retina in depth, we see, which again, it's kind of this like back area of the eye. We see that there are three main types of cells in the retina. We have the ganglion cells here, the bipolar cells, and then we have our photoreceptors, which are two types of photoreceptors, the cones and the rods, which we will go more in depth into that as well. But as you can see, light enters this way, and it actually kind of passes all these ganglion and bipolar cells before landing on these photoreceptors, which then triggers these chemical reactions in these photoreceptors, which in turn activates these bipolar cells, which activates these ganglion cells, which then converge together to make the optic nerve, where information is sent to the visual cortex. So it's really important to understand that, light kind of goes through and kind of has to figure out its way past all these cells before going onto these photoreceptors, and then output information goes back. And these three cells kind of just work together in a group to be able to process this visual information. So specifically looking at our two types of photoreceptors, we have the cones and the rods, which get their name based on their structure, as you can see here. And the cool thing about the cones is that it deals with color vision, where the rod is kind of known for night vision. So it doesn't deal with color, only the cone deals with color. And therefore, the cone kind of has like some higher acuity and faster transaction pathway, because when we're seeing light, we got to be able to process that light information very quickly. And The higher acuity in the cone comes from the fact that there's like one cone for every one bipolar cell, where in rods, there's lower acuity because there's hundreds of rods to one bipolar cell. So the information that goes onto these bipolar cells is different, and is slower in one and faster in the other. Which when you think about how you see in light versus dark, it makes sense why cones have to be fast and they have higher acuity compared to in dark. You turn the lights off, it takes a second for your eyes to process what you're seeing. You can't see as well. Or in light, you're processing things very fast and being able to see things in great detail. These two photoreceptors dominate different areas of the retina, which I have a great image that kind of shows the density of these rods and cones, but cones dominate the fovea, and there are no rods in the fovea. And the fovea is this very important area of the retina, which allows us to see things in great detail. But rods are located in the periphery, and there are cones in the periphery as well, but way less of them. So these two photoreceptors dominate different areas of our retina. And you can kind of see this here, but this is the fovea. This kind of like dip area is the fovea. And here again, we're looking at the retina. So it's the ganglion cells, bipolar cells, and our photoreceptors. And then this area, it's essentially an area that can absorb scattered light. So it makes it easier that if light is not kind of going anywhere, this bottom layer will absorb it so that we can focus on things better. But the really cool thing here is that the ganglion cells and the bipolar cells move out of the way so that light can directly go to the photoreceptors and not have to, find its way through all these cells. And that is one really important piece to the fovea, and one reason why it allows us to have such high acuity vision here is because the light can directly hit the photoreceptors. And again, these photoreceptors down here are only cones. There are no rods seen here. And so this is just a really important part of the retina that allows us to see things in detail, see colors in detail, and allow for high acuity vision due to its high density of cones and due to the fact that these cells will move out of the way so that light can directly come onto the cones and the photoreceptors there. And this is a great image that kind of just shows here. We have the fovea here. So you can kind of just see that there's only cones and there's a very high density of cones. And then as you step out of the fovea into the periphery, there's rods, and the density of rods decreases as you move away from the fovea. So just really cool that our retina kind of structures that way and changes the density of these rods and the cones so that we can view things, kind of just optimize our vision, which is really cool. And in the human, we have 5 million cones. And as you see, that large density in the fovea, and then there's 100 million rods. So way more rods and cones. But where they are at in the retina is different. And kind of a fun fact about the retina too is that we have this blind spot near it as well, which is just interesting and kind of cool to think about how we have a blind spot that is near such important structure for being able to see things. But one important part about the fovea too is that because it has such a high density of cones and such high sensitivity, it's represented a huge amount in our cortex in the brain, which just kind of leads to this idea of retinopathy and cortical amplification, that something that has high density and a big importance in one part of the brain, or in this case in the retina, is going to also have a high density and a big impact in another part of like the brain. So this just kind of leads to the idea of retinopathy, which is seen a lot in the visual system and other systems in the brain. So kind of going back and taking a look at things on the greater scale after we've kind of been able to understand how light coming in is viewed in the eye, and being able to be absorbed in the eye, to go on to do other things. After we've kind of been able to understand how that happens, going back and looking at how it leads to our primary visual cortex and our occipital lobe is interesting as well. So here we have the right visual field and the left visual field. And this left and right can be kind of confusing, but if you imagine yourself standing here and looking this way, you're going to see that the right to you is these blue and the left to you are these red. But as we see, what we see in the right visual field is then processed contralaterally in the left hemisphere. So when it crosses kind of over into different sides of the brain, we call this contralateral. And we see the same thing here, that the left visual field then crosses over and is kind of processed by the right hemisphere. And here we see the optic nerve, which we talked about, those ganglion cells converging to and heading out of the retina. We see that optic nerve coming back, and it is processed by this area called the LGN, which then takes the information to the primary visual cortex. The LGN can be viewed here. So this is kind of like a close-up of the LGN. And we kind of see that, again, it serves the contralateral visual field. And here we see different types of ganglion cells. We see these, we call the M channel, these M cells, M ganglion cells, which are larger, they're faster, they respond to movement, they're mostly rod input, and they perceive like differences in brightness. And then in these P cells, we see that they're smaller, they're slower, they can respond to like specific attributes of stimulus, and they're mostly cone input. So these P cells are kind of more focused on color, texture, fine detail, which corresponds to why it's cone input. But as we see here, there's again this like retinopathy in the way that the spatial organization of neurons as they travel like stays together. So these, P cells are gonna stay together, and these M cells are gonna stay together as they go onto the LGN, and they do this contralaterally, and they end up in different layers of the LGN. And then from there, the information from the LGN then goes to the primary visual cortex. So the primary visual cortex can be seen in this green area. And this whole area is the occipital lobe, and then we have the temporal lobe, parietal lobe, and the frontal lobe. And so our vision is focused, starts back here. So you go from the eye, you're going back to the primary visual cortex, where then the primary visual cortex will send to other areas of the brain. And then we call this the secondary visual cortex, just the rest of the occipital lobe that's not in the primary visual cortex. So there are different visual cortices related to this occipital lobe. And the V1, it's kind of where it all starts, which is the primary visual cortex. And then from there, it goes to different areas. And all of these different cortices are like known to focus in different areas. So V4 is color and form. V3A can deal with motion. V2 relays signals. So we'll see V2 show up a lot. So all of these different cortices kind of have something that they specialize in. And there is known to be 8. And yeah, this is kind of a better view of how they align in the occipital lobe. But looking at how information is kind of spread from this primary visual cortex to these different areas, this does a great job at kind of giving an overview of how it goes through. So you can see like right now we have the M cells, P cells, the LGN are those like M layers and P layers that I showed you with the cells. And then from there, it goes to the primary visual cortex, which is this V1. We can see this here. And then from there are 2 different pathways that then send information to different visual cortices. So we have the dorsal pathway, which is, we give the analogy that a dolphin, the dorsal fin is on the back, kind of goes up, and this is going to go kind of to the direction of the parietal lobe, where we have the ventral pathway, which is underneath, which is going to kind of go to the temporal lobe. So looking at these pathways, specifically the dorsal stream is known as, as the Ware pathway. And this pathway leads from the primary visual cortex to V2 to V5, mostly V5, and then also V6. And this, again, is that upper stream, so it's going to lead to the parietal lobe. And this dorsal stream focuses on localization of objects in space for action-oriented behaviors. So if you think about a lion running at you, this can allow you to move away from the lion, or if a ball is being thrown at you, you reaching your hand out to grab the ball. So that's what this kind of like area focuses on, is when you see something, how can you then move your body in response to that. So damage in this area can cause an inability to voluntarily move, and an inability to like perceive motion. Yeah, and then the ventral stream is known as the wet pathway. So this goes from the primary visual cortex to V2 and V4, which leads to the inferior temporal cortex, which is just kind of like the inferior bottom part of the temporal lobe. And this area deals with object identification and recognition, hence the wet name. This is long-term memory storage. So a lot of this deals with like faces, your ability to remember people you've met and faces, where the Ware pathway is not based on memory situations. So damage to this area can cause lack of facial recognition, inability to recognize facial expressions. This area and its known like ability for facial recognition is a very interesting topic and very cool to learn. So I'd recommend looking into that if you're interested, because it is very interesting. But yeah, these 2 ventral streams kind of allow us to be able to our brain to process what we're seeing and respond in different ways, which is just something really cool and it goes very in depth, and it's really interesting to think about that we do these things without even thinking about it, which is just such a cool part of the brain. So with all of this, I know last week we talked about computer vision and some similarities, like the brain analogy to computer vision. So one similarity is that neurons at different levels respond to increasingly more complex features, just as layers do in CNNs. This was talked about last week in lecture, and there's some like couple good, I think there's like 3 or 2 slides on this that I would recommend looking back on now that you have a better understanding of how the human visual system works. And then another similarity that is kind of talked about a lot is like the use of receptive fields. So in humans, neurons respond to specific regions of the input data, just as in CNNs, the convolutional layers use filters or kernels to scan receptive fields across the input data. There's very other similarities that I know individuals talk about, such as like the weights and different things like that. I honestly don't feel confident in my knowledge of CNNs to specifically make big statements tying it back to the visual system, but there is a lot of great research papers out there and a lot of work in these fields and the intersection between these two fields that I would look into if you're interested. Um, but yeah, that's a little bit about the visual system and how we as humans are able to see and process information. So yeah, thank you for listening. If you have any questions, I know we're virtual this week, but don't hesitate to reach out to me on Discord, and I will try to answer those questions that you may have. But yeah.